is Maya Chopra, who is the Rosamond Stone Zander Translational Neuroscience Center Director of Translational Genomic Medicine. Did I get that right? And please welcome her. There'll be time for questions at the end of the session, so hopefully we'll have a little time to do that.
Well, good afternoon, everybody, and welcome to track 4. I'm absolutely delighted to be opening this wonderful track at this conference. And a special shout out and welcome to those of you who are new to this forum, who are new to rare disease advocacy, and especially to those who are feeling a little bit overwhelmed with some of the terminology and this kind of jargon that's being used. And I hope that this is just an introduction to introduce you to the flavor of the kinds of things we're going to be talking about over this afternoon and tomorrow in track 4, to familiarise you with those concepts. And maybe for those of you who are already quite familiar, it's a refresher. And for those of you who know everything and you're just here for the humour and the charm, you're going to be a little disappointed, and I recommend track 3. But let's get started. So the outline of this talk is in 3 parts. We're going to be going over some very basic genetics. And basic genetics is important not just because it's interesting for geneticists, but because understanding the genetic mechanism of a disorder is the first step to understanding therapeutic options. We're going to move into the discovery cycle, the building blocks needed for therapeutic development. And I'm going to use neurodevelopmental disorders as an example because that's my expertise. But I think a lot of those concepts are translatable to a range of other systems. And then we'll touch on some of the therapeutic options that you're going to hear more about later today and tomorrow. So just an introduction to give you a little bit of a flavour and I hope tie all of these concepts together. So let's start very basic about genes and chromosomes. So as most of you will know, genetic material is in the nucleus of every cell and packaged into these structures called chromosomes. And we have 23 pairs of chromosomes. In our cells, 22 are called autosomes. We get 1 pair from our mother and 1 pair from our father. And then we have the sex chromosomes, of course, X and Y. So a gene is a segment of DNA packaged on these chromosomes that encodes a protein. And a protein is a compound that performs a variety of tasks. Proteins have a whole range of functions. They may range from scaffolding, structure like collagen, which is very important for connective tissue and bone, or perhaps coding a channel, an ion channel in the brain, which is important for maintaining the right chemical status, which when disrupted can result in seizures, for example. And in my field, there are proteins that are important for controlling the expression of a whole range of genes, and they're the kinds of disorders where we might see a whole range of features across multiple body systems. We think about the genome as a cookbook, and chromosomes being like chapters, and a gene being a single recipe, a recipe to make something. So when we think about a variation in a gene, we think about a variation in a recipe that changes the end product. So DNA represents the words. And in this example, there's one specific instruction, add 1 cup of flour, and then we can look at the kinds of examples of variations in those instructions and what happens. So you've heard a little bit about the concept of mutations or variations in genes, and these are just some of the examples of the types of variations that we come across. And this is important, again, not just because it's interesting, but because it informs the kinds of treatment options that may be available for genetic disorders. So in the first example, that add 1 cup of flour has been completely cut off after the U. The sentence stops too soon. And in that sort of situation, in most cases with a nonsense or a truncating variant, you end up with not enough cake or no cake, depending on whether that variant is on 1 or 2 alleles. In missense, so we have a spelling mistake, and a missense variant can result in different kinds of effects on the cake. Then you have an example of an insertion or deletion, so an extra letter or a deletion of a letter. Again, it can change whether that cake's going to be not normal or whether it's going to be less cake, and similar for duplication. And another example is splice site, and that's something you're going to hear about later today, especially when we get to the ASO section. So I'm just going to touch on patterns of inheritance. Again, we think about patterns of inheritance because it helps us inform the mechanism of a disorder. So this is an example of an autosomal dominant pedigree in which there are affected males and affected females. So typically this is the kind of disorder that can be very variable. An example that we study at the Rosamond Stone-Zander Translational Neuroscience Centre is tuberous sclerosis complex, TSC, which is an autosomal dominant disorder resulting in autism, epilepsy, and a range of benign growths through various body systems. And we see a lot of variability in TSC. So these children may present quite severely, and sometimes we find a parent's affected but much milder, but they have the same variant. This is very typical of autosomal dominant disorders. So just to remind you about the genetics, these are on autosomes, where we have one copy of the gene that's standard and one copy that has one of the many types of mistakes, and there's a 50% chance of inheriting each of those options. And then, of course, more often than not, in my field, in neurodevelopmental disorders, we don't quite see that sort of pedigree. We see it starting in an individual, in a child, with an event we call de novo, so starting just in the egg or in the sperm that made up that individual. So for autosomal dominant disorder, there's a 50-50 chance, 50% chance of inheriting the standard copy, 50% chance of inheriting the copy that has the spelling mistake. So a 50-50 chance, tossing a coin, of being affected with the disorder with each pregnancy. So autosomal recessive, we heard an example of an autosomal recessive disorder this morning, of course, with Niemann-Pick disease, where you are okay if you only have one copy that's normal. So for example, parents are carriers, that means they have one normal copy, one with a change, and they're fine. They have no symptoms, they're absolutely asymptomatic. If the child inherits both of the mutated copies, then the child will have the condition. And that's a 1 in 4 or 25% chance. And more often than not, these recessive disorders are what we call biallelic loss of function. That is, both copies of the gene are mutated, have been wiped out, so we don't see any of that protein that we need for that particular function. X-linked recessive, so an example of this might be, for example, haemophilia, where we have girls who have 2 X chromosomes, so if there's a mutation on a gene on one X chromosome, they have a backup copy. So typically they're asymptomatic or maybe have mild symptoms. Males, of course, only have 1 X, so if that has a change on the copy, they don't have a backup on the Y, so they are affected with the disorder. So for X-linked recessive conditions, if the male is affected, the parent, all of the females will inherit that X and they will be carriers. But all of their boys are fine, of course, because they will inherit the Y from the father and the healthy X from their mother. For female carriers of X-linked disorders, there's a 50% chance of passing on the healthy X, 50% chance of passing on the X that has the change. So for those, 50% of their sons will be affected with the disorder and 50% of their female children will be carriers. So those are just some examples of some patterns of inheritance, and I think you will hear about options for treatment like upregulating the wild type copy or replacing a gene that's missing. And I hope that it will help you to have a little bit of context that some of those options apply to some but not all of those patterns of inheritance. And of course there are many other types of disorders that we haven't discussed. X-linked dominant conditions like Rett syndrome and CDKL5 deficiency disorder, where girls actually are affected because this gene is very important in terms of dosage. And the boys are either more severely affected or so severely affected that it's lethal at birth. We haven't talked about imprinted disorders. These are disorders in which it's very important to know which copy has been inherited from the mum or the dad, because that determines whether the gene is switched on or off. So when you hear about disorders like Angelman syndrome, Prader-Willi syndromes, these are disorders of imprinting. And I think all of this is relevant because we often have advocates come to us and say, well, this treatment really worked for such and such disorder, but actually it's because of very specific mechanisms that made it possible for that treatment to work. Chromosomal disorders, of course, aneuploidy, chromosomal microdeletions and duplications, changes in the number or the structure of chromosomes. Very important, especially again in my area of neurodevelopmental disability, an important contribution to that burden of disability in our community. And mosaicism, so having some cells that are affected with the disorder and some cells that are not. And we see that a lot in our vascular malformation communities. And of course, just a nod to remind you that I'm interested, a lot of us are interested in monogenic rare disorders, but of course, there are many rare disorders which are influenced by genes and lifestyle and the environment. So in the US, as many of you will know, a rare disease is one that impacts fewer than 1 in 200,000 people. This is a huge worldwide burden of 300 million people worldwide affected by rare disease. There are over 10,000 rare disorders, and 1 in 10 people in the US are affected by rare disease. And of course, what this tells us is that collectively, these are not rare. This is a common and significant public health issue, and we're all here to shine more light on that. This is from the Global Genes website. I think many of you are familiar with this, but the one thing I wanted to highlight is that 4.8 years, the average time to make a diagnosis for rare patients. So we only know what we know. Many of you have children, family members with rare disorders. There are many people who couldn't make it here, who are unable to, who are also affected with rare disorders. And then there are those who have never even reached a diagnosis, particularly underserved communities. So just a little nod to those people out there. So, and this is maybe not tapping into the birds of a feather session that is coming up later on this week. But we can think about rare diseases individually, or as you connect with each other, you'll start to think about how you can group. You might group them by organ system, so I think a lot about disorders that affect the brain. You might think about cellular pathways. So for example, you might think about genes that control transcription, or ion channels, or synapses. Or you might think about genetic mechanism. What is the root cause? For example, you might think about a mechanism like haploinsufficiency. So those autosomal dominant disorders where 1 copy of the gene is missing and 1 copy of the gene is present. So we're just going to turn our attention now to my area of interest, neurodevelopmental disorders. So for rare disorders, 70% have a childhood onset, and of those, 90% have neurological or neurodevelopmental impact. And you all know, and this is why we're here, there's a major gap between the identification of genes, which has really exploded over the last 10 years with the genomic revolution, and the availability of therapies. And something we hear very often is, well, we know what the gene is, so why can't we just fix it? And I hope that this talk will give you a flavour of all the different steps that need to happen before we can get quite to that spot. So using neurodevelopmental disorders as an example for the discovery cycle. It all starts with defining patients, in this case neurodevelopmental disorders, characterising the phenotype, truly understanding the disorder and its natural history, and understanding biomarkers, and we'll get to definitions of some of those terms. Developing preclinical models, for example, mouse models, or we can model neurons using patient-derived cells, which we can transform or reprogram into neurons to understand how they're different, and using those to test therapies for safety and efficacy, for example, in a mouse model. And all of this leads to clinical treatment trials, which then feeds back to better understanding the disorder. And sometimes this takes a few cycles. And I really do like this slide. I use it a lot because I think it's a reminder that the process is not linear. It's a cycle, and often we have to go through it a couple of times. So the big question is, why does research take so long? Sad face. And I set that up, but I worry that it looks like I have the answer on the next slide, but I don't. But really, I just wanted to be mindful that we hear you and we get it. And you know, it does take long. And a nod to the beautiful roadmap developed by Global Genes, which really highlights the try-agains and the turnaround and the curvy nature of this road and this map that we all want to get through. So I'm just going to touch on a few of the flags, concepts, phrases and words you're going to hear throughout track 4. So let's talk about preclinical validation. So what does preclinical validation mean? It means having validated preclinical model to show that the tools, whether it be a vector or an ASO, get to the target organ, be it the brain or the muscle or the eye, at the right time. So what does the right time mean? It means you have to know what the temporal expression window is. That is, what's the window of time where expression of that gene is super important, because you need to get in before that. And then to be able to demonstrate reversibility in a model system, to show that if you get in on time, you can reverse the phenotype, and then obviously to be able to test for safety and efficacy. Some examples: AAV is a vector for gene therapy, so that's a delivery tool, and then an example of reactivating a gene for a disorder called SCN1A, haploinsufficiency disorder, demonstrating that if you reintroduce this gene, when the mouse already has a phenotype, you can reverse it and make the mouse better for Dravet syndrome. I'm not going to talk too much about this slide, but just to recognise that many of you will be thinking about preclinical models and deciding between mice or iPSCs, and how do you make that kind of a judgment. And obviously there are pros and cons for both, but ultimately I think they are complementary model systems. So for example, if we look at the cube, which quite nicely demonstrates these 3 axes: human relevance, mechanistic insight, and throughput. So if you think about mice, for example, they have a high mechanistic insight. There's a lot of things you can model in a mouse, like behavior. You can't model that in a cell. Human relevance is I guess, less relevant, and the throughput is intermediate. In contrast, you have iPSCs, excellent human relevance, excellent mechanistic insight, but the throughput, this actually slide was taken from a journal article a few years ago, and I apologise, the reference has dropped off, but I think the throughput is moving deeper. I think it is getting better, but obviously this is still a very costly preclinical model to develop. So clinical trial considerations. So we've talked about the genetic mechanism, we've talked about preclinical, and now we're going to talk about clinical trial considerations. So we have the concept of a contact registry versus that of a natural history study. A natural history study allows us to understand the disease course, the spectrum of severity, the range of manifestations, and to ideally identify the subset of the population most likely to respond to treatment. And we think a lot about what's the typical diagnosis of a disorder versus the temporal therapeutic window, meaning if we know that you have to get in and treat a disorder by the age of 2 in order to reverse the phenotype, but we also know that for this particular disorder, patients don't get diagnosed until they're 5, well then we need to redirect our efforts to getting patients diagnosed earlier so that we can even have a chance of getting into that really critical temporal therapeutic window of opportunity. So just to expand on a registry versus a natural history study, a contact registry allows patients to connect to clinical trials and to other research opportunities, and sometimes to each other. And these typically include individuals with a specific disease who are interested in enrolling in future clinical trials and want to be contacted. And some of that data may include contact and demographic information and some health information to determine clinical trial eligibility should those opportunities arise. And then, in contrast, a natural history study informs every part of drug development and benefits the whole disease community, typically containing detailed, ideally longitudinal, as opposed to cross-sectional, so at multiple time points, age of diagnosis, features, symptoms, test results, medical images. And hopefully some of that data from natural history study will help inform the endpoints which will be measured for when you get to the point of a clinical trial. Data may be entered by patients, it might come from the EHR, it might come from clinical assessments, it might come from validated standardized research assessments, or directly from patients and caregivers. And I think you will hear some really robust discussions about the types of data we should be collecting in natural history studies throughout this meeting. Clinical trial endpoints are absolutely crucial to trial success. We need them in order to succeed in clinical trials. So we talk about endpoints as being direct, how patients feel, function, and survive, versus indirect, such as biomarkers, for example, a blood test or an EEG. The endpoints need to be meaningful to the patient population. And one thing you're also good at doing, I think, as advocates, is connecting with your communities to feed back to us as researchers to say, this is what's important, because it's not always intuitive. So, for example, a researcher might think, well, seizures and epilepsy, control of epilepsy is the most important thing, but families might come back and say, actually, communication is really important. That's the most important thing. We just want our child to be able to say what's bothering them if they're hungry or they're in pain or they're distressed, because that would really make a difference to our quality of life. And I think that's so, so important, and we love to hear your voices to understand what are the important endpoints and how can we then measure them. We have trouble in the neurodevelopmental space because cognitive endpoints are really hard. It's really hard because the way that cognitive measures have been designed is for a normative population. So it's very hard to apply those measures to children who have neurodevelopmental disorders. In contrast, survival motor endpoints, surviving, walking, being able to sit, they're very nice, concrete measures that are so much easier to design a clinical trial for. And perhaps that's why we've seen such success. One of the reasons we have seen such success with SMA, with nice curves like this one demonstrating a clear survival advantage for those who are treated with gene therapy. Those who are able to sit and stand. These are very clear endpoints and much, much harder for us to be able to validate neurocognitive measures in this concrete way. This could almost be a whole talk in itself, ethical considerations. It could almost be a whole conference. So I hesitate to try and cover everything, but I also want to acknowledge how important it is. It's only ethical as researchers that we engage with you all, with families. It's ethical that we think about risk versus benefit, not only for a disorder, but for an individual, a family sitting in front of us and making that decision about whether or not to enroll in a clinical trial or not. And we talk about this a lot. What does a cure actually mean? And that's up to you all to tell us so that we can hear you. What does a cure mean? What are we actually looking for? And then I guess one of the really big things that I think about that keeps me a little bit awake is how are we going to ensure equity of access to research opportunities and to treatments, both within the US, where we know there's such discrepancies in access to health and access to research, but even globally, how do we even think about those concepts? And again, just to recognise the work from the FDA, who acknowledge the importance of drug development as being patient-focused. You're all much better at me than thinking about where do we even start with funding. So of course, these are the options for government, private companies, non-profits, philanthropy, all of which I know you're all deep in discussions about. So now we're going to turn what we've learnt from genetic mechanism, preclinical, clinical, to kind of talking about therapeutics. And again, you're going to hear about these individually throughout the course of this afternoon and tomorrow. So this is just to give you a little appetizer, or entree, as we would say in Australia, which for you means main course. So we went straight from DNA to protein, I think, in the last slide. So there are a couple of extra steps there. So DNA genes are made of exons and introns, exons which are coding, introns which are not coding, and they get spliced out when DNA is transcribed into RNA, which then gets translated into these building blocks, these amino acids, which then fold and undergo post-translational modifications to make the protein that has a job to do. And so when you hear this afternoon and tomorrow about various therapeutic options, you can think about this as a roadmap and being able to intervene at different points depending on the therapy. So, for example, when we talk about gene therapy, we're talking about replacing the gene, the DNA. Ensuring that the correct gene is expressed. This is generally a one-time therapy. In my field for neurodevelopmental disorders, we talk a lot about the difficulty of delivery to the CNS, to the central nervous system. We talk a lot about the size of the gene and whether it fits into a vector. There's size limitations for AAV vectors, and a lot of the really critical neurodevelopmental genes exceed this size. We think about the immune response and the challenge and the expense of manufacturing. And just a nod again to spinal muscular atrophy, which we'll be hearing about later, and the success of delivering SMA, replacing the missing gene, which is called SMN1, needed for the SMN protein, and importantly, being able to deliver that to the target tissue, the anterior horn cell. So then we move on to intervening at the point of RNA. And typically this requires multiple doses. This is where you hear about terms like antisense oligonucleotides. These agents do not cross the blood barrier, so the blood-brain barrier, so they need to be infused directly into the cerebrospinal fluid. And they can be used to decrease expression, or actually to increase expression, or to modulate splicing, depending on the underlying genetic mechanism. An example here is an ASO that's developed for Duchenne muscular dystrophy. So in this example, on the left, there's a deletion in exon 50, which seems to result in some premature truncation or a premature stop in exon 51, resulting in a protein that's non-functional, it's truncated, doesn't work. By treating with an antisense oligonucleotide, binding to exon 51, we can change that type of mutation to another, where exon 51 is out, but at least the reading frame continues, and you end up with dystrophin that's not quite right, that's missing a little bit, but it's functional and it's going to do better than the mutated copy. And this is now exon DYS51. Is now in use on compassionate grounds. And I think about 15 to 20% of patients with Duchenne muscular dystrophy are eligible for this kind of treatment. And then small molecules, again, an advantage with price, scalable, and some are already FDA approved, which is a huge advantage. And that's where we talk about drug repurposing. There may be off-target effects. And again, we're looking at in this sort of cycle, impacting downstream of the protein being produced. So with small molecules, we're not changing the gene, we're not doing anything to the RNA, we're helping the protein do its job. An example is rapamycin, so for tuberous sclerosis complex, that autosomal dominant disorder we talked about. It's due to a mutation in either one copy of TSC1 or TSC2. And these are, we think about TSC1 and TSC2 as brakes to a growth pathway called mTOR. And when mTOR is overactive, we get epilepsy, autism, and these benign growths. Cell proliferation is how it's described in this slide. And it just so happens that there's already an mTOR inhibitor that's been FDA approved and that was used for, as a drug for following renal transplant to suppress the immune system. And so this has now been approved, of course, for multiple indications in tuberous sclerosis complex. So let's just talk a little bit about genetic mechanisms, loss of function versus altered function. So on the left, you have two copies of a gene producing a protein, an orange protein, let's call it. It's normal, both copies are working, it's producing plenty of the protein. And then we'll say there's a variant or a mutation on the lower copy, so you've only got half of the protein being produced. So that's kind of a disorder we would call haploinsufficiency. That's the type of mutation. Then we have on the right, we have again the same standard example. And then when there's a variant, instead of making orange protein, that copy of the gene is making black protein. Now that protein might go off and do something completely different. It might actually interfere with orange protein, so cause a dominant negative. Or it might just be an overactive, hyperactive version of orange. So it's getting too much orange. And this is so important because it speaks to, again, if we're starting to propose therapies, if we're saying, well, why don't we just get the top copy to make more orange, we need to know what the black protein's actually doing before we start heading in that direction. So I hope that explains why we get very focused on, is this loss of function, is this gain of function? Goldilocks, so it has been shown that most genes are actually bidirectionally dosage sensitive. We need just the right amount in order to be healthy. An example being MECP2, which results in an X-linked disorder called Rett syndrome, which we referred to earlier. And that's girls who don't have one of their working copies of MECP2. But of course, we know that having too much MECP2 from children who have duplications or extra copies also result in a syndrome. And this is very important because if we're talking about, oh, not enough MECP2, let's replace it, then we need to think about, well, we don't want to replace it too much because we may actually end up doing harm. So I'll just pause for a second. We've talked about a lot. We've talked about genetic mechanism, we've talked about preclinical, we've talked about clinical, we've talked about ethical. And our group, the Rosamond Stone-Zander Translational Neuroscience Centre, felt that there was a real need to put all of these considerations together. And so that's what we did. We wrote this paper called Gene Target, a framework for evaluating Mendelian neurodevelopmental disorders for gene therapy. Again, we've written this for neurodevelopmental disorders, but I do think a lot of these concepts are relevant for other disorders. And the idea is that each letter of this mnemonic represents a group of considerations in those 4 categories, and that those could be used for PAGs to think about where should we allocate our resources, where are our gaps. For example, we understand the genetic mechanism and we've got a preclinical model. What we don't have is endpoints in natural history. So to find the gaps to which to direct your resources. And then we hope it's also useful for clinicians and scientists to look at a disorder and to say, well, where are the gaps that we should focus on, and how do we prioritise which disorders are ready, which disorders should we that really push through for clinical trials. So just to show you what the mnemonic looks like, and I would encourage you all to read the paper. And really it's a summary of most of what we've talked about and a lot of what you'll hear about in the meeting. So genetic mechanism needs to be understood and amenable. So we need to understand the genetic mechanism, not just know what the gene is. We need to know, is it recessive? Is it dominant? Is it gain of function? Is it loss of function? Is it a mix? What are the missense variants doing? What's the full range of the genotypic spectrum, and are we capturing them all? What do we do with variants of uncertain significance? Are they relevant? All of that falls into genetic mechanism and is really foundational for therapeutic development. Early diagnosis is typical, and I wish that, and I hope that we're going to soon live in a world where early diagnosis is typical for every rare presentation. I mean, this is critical. It's critical to get those patients diagnosed early, even when there's no treatment available, because that's how we're going to collect the right amount of data in order to be able to push forward for therapies. We need to understand the natural history, the breadth, the longitudinal natural course, the expected course of that disorder, so that we can understand when you intervene, what does that mean and how does that change the trajectory? We need to have validated and meaningful endpoints. We need to have tools that deliver to the target tissue at the right time. And so implicit in that is we need to know what the right time is, what's that window of therapeutic opportunity. Availability of other safe and effective treatments is limited. What we mean by that is we should prioritise disorders for which there are no treatments, which is most. Reversibility has been demonstrated in a model system. So if we had, for example, that mouse model where we can reintroduce the gene or reintroduce expression and we see a change, that gives us proof of concept that the disorder can be reversible. The gene is tolerant to dosage changes. We've already said most genes are not very tolerant, but at least we should know. We should know what that tolerance, what that dosage window is. Ethical principles have been considered, again, at an individual level, but also at the level of the community and the level of accessibility, and making sure that these treatments can get into the hands of the global rare disease population. And target population is engaged and accessible. And again, this speaks very clearly to you all as being here and being advocates for your families and for each other, to want a treatment and to know exactly what it is that you want out of a clinical trial. So to summarise, thinking like a researcher, I think that it starts with being able to articulate a scientific question. I think that you and your scientific advisory boards are in a good position to identify the gaps in your translational pipeline. And where the magic happens is when you can find a researcher who has a scientific question that matches your translational gap. So if your translational gap is we don't really know what the genetic mechanism is because we have too many missense variants and variants of uncertain significance, find a researcher who is interested in answering that part of the question. There's a healthy tension in timelines, and we talked about this in Philadelphia. We know the urgency and we hear the urgency, and we need to continue to feel that from all of you. But at the same time, we need to be responsible and we need to be purveyors of good and sound science and safe clinical trials that don't fail, because that's what we don't want. And then the pathway to therapies start with these very basic building blocks. We all want to get to the finish line, but we need to sort of build the foundations so that we have the best chance of succeeding. So I'd just like to recognise that my talk is a reflection of work of the entire Translational Neuroscience Centre executive team, particularly recognising Mustafa Sahin and Kira Dyes, who is here, and you may wish to join us at our panel tomorrow. Recognise my team, the Translational Genomic Medicine Core. Ray Belanger is here, and he is our new community outreach coordinator, so please feel free to speak to him. And thank you to all of the patients, the families, the parents, the enrollees in research studies that I've come across. We see you, we hear you, and we're inspired by you. Thank you.
Hi, Steve Grossman. I represent the ADCY5 movement disorder community, and our gene is a gain of function, and it's rather large. What do you see as some of the options or possibilities coming down the pike for large payload genes like that?
A gain of function, dominant disorder? Yeah, I wonder about maybe an RNA-based strategy to downregulate. But again, if I were approaching this from a translational standpoint and the building blocks, I would think about dosage and what happens to people who have one copy of the gene and whether there's been any documented and the full mutational spectrum. Gain of function, I mean, you know, it's just very hard. It's very difficult to prove that it's not dominant negative or that there's an altered function. So I would start with those building blocks, and then working towards maybe thinking about downregulating the dose. Yeah, and I think we're seeing more of these sorts of disorders, and we're seeing more of one gene, 2 or 3 mechanisms. You know, so a disorder might be described initially as a haploinsufficiency disorder, and then the missense variants start to appear, and they're often a mix of loss of function and gain of function. And I think that that's going to be one of the big challenges of therapeutic development as we move forward. And we start to see kind of more of these one gene, multiple mechanisms, multiple overlapping disorders with overlapping phenotypes.
Up front, Shruti. I can ask you a quick question.
Please.
You had your magic cube.
Oh, yes. Yes.
So the iPSC cells, and you had the mouse models. What about organoids? Are you seeing that as something different than iPSC cells? And, you know, is that giving, where would you put that in the cube continuum?
Yeah.
And is that giving more value than either of those two?
Yeah, very good question. I think certainly for our group, so I think we're working towards developing organoids within our human neuron core, but where the concerns are with really reproducible protocols to build those model systems. So we're already struggling with reproducibility, even just for iPSC lines and differentiating into a single neuron type, and seeing getting consistent, reproducible, valid results. And I think that is even more challenging for organoids. So I would say it's in development. There are others who may have more expertise in that field, but definitely an area to watch.
Hello, Doctor. In the next 5 years, what do you think will be the major breakthroughs in gene therapy, particularly related to the brain?
You know, I can tell you what I'd like to see. You know, the things that we keep coming across are getting patients diagnosed, getting a delivery mechanism that can deliver to the brain and specifically to the target tissue. Delivery is a huge problem. Manufacturing. And then I think the broader kind of community considerations about who's going to be able to access gene therapy and how is that going to be resourced. These are rare disorders. Some of them, there may be only a few patients in the United States. We're going to have to start to think globally, and we're going to have to leverage our registries internationally, which then leads to the question of how we're going to actually make this work across multiple different health economies and health systems. I think they're I feel like they're sort of the sorts of things we'll be talking about in the next 5 years. Yeah, really good question.
In the back?
Hello. Thank you for your presentation. You had the slide with the 2 copies of a gene and the orange dots, right? Like on the left side, it's like one is producing a functional copy, the other one is not producing anything at all, and then on the other one is functional and the other one is altered. And you mentioned the term haploinsufficiency. And are there any known successes in therapeutics that approach the solution by making the copy that actually works, that is functional, somehow making it work double to make up for the one that is non-functional versus trying to address the non-functional copy?
Yes, yeah, another really good question. Yes, the successes have been in preclinical models, for example, in disorders like Dravet syndrome, but then being able to have that proof of concept in a preclinical model is a starting point, and then the bigger challenge is what we've talked about in terms of getting those patients diagnosed and having endpoints in order to move through to a clinical trial. Haploinsufficiency in, going back a step, the mechanisms that cause neurodevelopmental disorders will depend on the population you're looking at. So in the US population, haploinsufficiency is a very significant and major cause of neurodevelopmental disorders. And I think the type of concept that you're talking about, upregulating the wild type copy, maintaining the isoform integrity of that cell, and being able to do so at the right time, I think that's actually very promising. And I think we'll see and hear about more of those kinds of treatments in the future. But so far, I think it's just preclinical.
We have quick time for one last question up front. Oh.
Thank you, Tim. Tim Stewart with the Shine Foundation. DLG4 is our gene and produces the protein PSD9D5. In the 85% of our population is haploinsufficient, but amongst that population of haploinsufficient cases, there's a huge, a very wide range in phenotypes. Is that typical with other rare diseases?
That's very typical for dominant disorders. So for example, tuberous sclerosis complex, to see that really wide range of severity and of spectrum of manifestations. And we may put that down to background genetic risk, epigenetics, environmental factors, genetic background, but ultimately I don't think we completely understand that. A disorder that I'm very interested in is caused by haploinsufficiency of a gene called ankyrin D17. And we have, in our research study, a pair of identical twins. So not only do they have the same ankyrin D17 mutation, but they have the same genetic background. One has a severe intellectual disability. She's completely dependent on her parents. The other had a bit of speech delay, had a bit of help with her speech, and she's now a real estate agent. So
How do you explain that?
I don't think we can. It's epigenetics. Is it perhaps we should do a genome on both of them and look for post-zygotic changes, post-zygotic mutations or copy number loss. Perhaps their intrauterine environment might have been at the same time, but it was different. Maybe one had an advantage in terms of cerebral blood flow. I actually don't know. I don't know. But it's super interesting because if we're trying to create a world where we can look at a mutation and predict, well, this patient's going to be severe and we need to get this one in a clinical trial, how can we do that when they're so variable? Yeah. So I hear you, but I don't know.
Please join me in thanking Maya. It was a great talk to set up what follows.
Thank you.